afternoon, dear students. I congratulate you on the start of your new academic semester. Today we have the last lecture from the genetic cycle of the medical biology, and after that we will begin to study parasitology. The theme of our lecture today is diagnostic and treatment of genetic diseases. What are the difficulties in studying human heredity? This slide lists the main difficulties in the study of the human heredity. Let's look at it. The first cause is huge genetic diversity, or in another word, genetic polymorphism, and genetic load variations. The second cause is slow generational change. It's about 20 to 30 years. The next point is a small offspring, and hybridological and statistical methods are not acceptable in the human populations. And the last cause is exceptional rarity of living condition. So what different diagnostic methods of hereditary diseases are known in our time? Let's see. There are many different type of the diagnostic methods, such as family history, or you can take a term genetical methods or family tree methods. Population statistical methods on the basis of the Hardy-Weinberg principle. Cytological testing, cytogenetic testing, biochemical testing, DNA diagnostic, twin study, dermatoglyphics, hereditary pathology modeling. And about all of these methods we will talk on this lecture. So let's continue. About family history or genealogical methods and population statistical methods on the basis of Hardy-Weinberg principle, you will talk some more and talk in the detail on your practical voices. We start from the cytological testing. So cytological testing, what is it? Cytological testing is determination of X chromosome, or in another word, Barr body. Another term in Barr body. A Barr body, named after discoverer Murray Barr, is an inactive X chromosome in a cell with more than one X chromosome, rendered inactive in a process called lionization in species with XY sex determination, including humans. So, as you know, the genotype of the women is XX, and one of the X chromosome is active and another X chromosome is inactive. Or another X chromosome, Barr body, is inactive, or the genetic materials are present by heterochromatin, no euchromatin like in active X chromosome, but heterochromatin, inactive genetic material. So, how do you think the sex chromosome of men is XY chromosome, and how do you think X chromosome of the man is active or inactive? I give you some seconds to think about these questions. So, X chromosome of the man, active or inactive? and I give you the right question, the X chromosome of the man is active. So how we can see the Barbori inactive X chromosome have only women in the normal genotype. Only women with normal genotype have an inactive X chromosome or Barbori. In the normal man don't have an inactive X chromosome or Barbori. Men's have only active X chromosome and Y chromosome. So you can see it in the slide. In normal, only women have a Barbori, and it can be diagnosed with cytological testing. Normal men haven't a Barbori. You can ask me, is it some tests which can help to determination Y chromosome? Yes, it is. Some methods can determination the Y chromosome, and this type of testing is called Y determination chromosome. And in normal, only men have this type of the chromosome, and women without this chromosome. It's simple. What we can talk on the basis of the cytological testing. Diseases which can be diagnosed with cytological testing. One more. The normal genotype of the women 36XX, 36 chromosome with XX sex chromosome, and the normal genotype of the man is 36XY. So in normal, women have one Barbori. What the disease when women have not Barbori? We studied in the previously educational semester. So women without Barbori, which will have the syndrome which called Shereshevsky-Turner syndrome. Shereshevsky-Turner syndrome, and the genetic formula will be 35X0. So 35, the women lost one X chromosome, so no 36 but 35. 35X0 chromosome, Shereshevsky-Turner syndrome. Woman with copy of Barbori, the genetic formula will be 37XXX, and this syndrome is called trisomy X syndrome. Trisomy X syndrome. We think that the men in the normal have not Barbori, but what does it mean when the man has Barbori? The genetic formula will be 37XXY, and this syndrome is called Klinefelter syndrome. This slide lists the termination of Y chromosome. I said it some early. Normal men have a genetic formula 36XY, and when the study determine one more Y chromosome of the man, the genetic formula will be 37XYY, and this syndrome of the man is called Y polysemy, XYY syndrome, or in other words, Jacob syndrome. We said it in the previously educational semester about this disease. It also have a very interesting name, prison basketball team syndrome. Now let's talk about another test and another diagnostic methods of the human hereditary diseases. is cytogenetic testing. Cytogenetic method is a study of the karyotype. We can study amount and the structure of the chromosome. The object of the study is metaphase chromosome obtained from cells. As a rule, it is a lymphocytes cultured in artificial nutrient media. This method defines, how we said some earlier, amount and structure of the chromosome. The second, gender. And the third, chromosomal and genomic mutations. This is a very important slide. It is the stages of cytogenetic methods. The first step is taking material, as a rule it is the blood, for research. We're taking the blood to take a cell of the blood lymphocytes, because lymphocytes have a big nucleus, and it is simple to study its genetic material. The next stage is cell cultivation on artificial nutrient media with phytogamma agglutinin. What is a phytogamma agglutinin? Or in another word, PHA, is a lectin found in plants, especially certain legumes. The lectin has number of effects on cell metabolism. It induces mitosis and affects the cell membrane in regard to transport and permeability to proteins. So, the artificial nutrient with phytogamma agglutinin helped to induce mitosis. After that, the next stage is colchicine supplementation. Colchicine is a drug which inhibits microtubules polymerization by binding to its constitutive protein tubulin. As availability of tubulin is essential to mitosis, colchicine may inhibit mitosis. As a result of its proteins, we have a biggest number of the chromosome in the stage of metaphase, because colchicine inhibits microtubule polymerization. And the next stage is metaphase cell accumulation. After that, next step is cell hypotenization. After coloration, and after that, drawing up an ideogram and its analysis. The result of cytogenetic methods is ideogram. This picture lists the ideogram. What is the ideogram? Is a diagrammatic representation of a chromosome, complement, or karyotype. The next method is biochemical testing. Biochemical testing or biochemical methods allows to diagnose only genes diseases. I remind you that the gene diseases is the same that metabolic diseases. The metabolism in our organism is a complex of different numerous of biochemical reactions. So these methods helped us to diagnose only gene diseases. The next methods of diagnosis of hereditary pathology is DNA diagnostic. The main method is polymerase chain reactions, or PCR. Is a method to rapidly make millions to billions of copies of a specific DNA sample, allowing scientists to take a very small sample of DNA and amplify it to a large enough amount to study in detail. And the next method is twins diagnostics methods. It is a very interesting method. So at first I want to remind you that the twins may be monozygotics and dizygotics. Monozygotic twins genetically identical. They look the same, they have the same gender, and dizygotics genetically not identical. They look like brother and sisters and may be different gender. Twins diagnostics methods. We must know a new term which called concordance. What is a concordance? Concordance is percent similarity in twins, mono or dizygotics. It's look like concordance monozygotic, concordance dizygotic twins in this formula. Concordance monozygotic, concordance dizygotics, concordance dizygotics. Knowing the concordance of mono and dizygotic twins by one trait, it can determine the contribution of hereditary and environment to the development of this trait. To determine the role of hereditary and environment in development of the trait, there are used the Holtzinger's formula or formula of Holtzinger. It looks like this, and let's see. H, it is a coefficient of heredity, and use the letter H, it means hereditary. H equally enumerating concordance of monozygotics minus concordance of dizygotics, and in the denominator 100, and it means 100% minus concordance of dizygotic. And the second part of the formula, it is a coefficient E. It means environment. The role of the environment in the determination of the trait. Coefficient E equally 1 minus H. 1 minus coefficient of heredity. And again, H, coefficient of the heredity. E, coefficient of the environment. And what conclusion can be said on the base on this formula? If the coefficient H equally 0.721, trait depends on hereditary. The trait depends only on the hereditary, nor environmental factors can't determine on the depend of this trait. If the coefficient of H equally 0.6, 0.5, 0.4, trait depends on both hereditary and the environment. Or in another word, we said trait with a genetic predisposition. Many numerous of the different diseases have the genetic predisposition. For example, schizophrenia, hypertension, ischemic heart diseases, and others. And if the coefficient H equally 0.3 or 0, trait depends only on environment. Different environment factors. As a rule, it is, for example, some infections, bacterial or viruses infections, this trait or disease depends only on the environment factors, bacteria, fungi, viruses, and others. Let's see on some example. The trait eyebrow shape. Concordance of the monozygotic twins is 100%, of the dizygotic twins 51%, and if we gave these numerals in the formula, we have that the coefficient H equal 1, and it means that the trait eyebrow shapes depends only on the heredity. No environment factors can't depend on this trait. At the same, eye color concordation of the monozygotic twins is about 100%, or the dizygotic twins is about 28%, and the coefficient of the hereditary one equal 1, and we can conclude that eye color it is a trait which depend only on heredity. And such diseases, schizophrenia, epilepsy, concordance of monozygotic twins is about 60%, and dizygotic twins about 20%. And in the result, we see that the coefficient of the H is about 0.6, and we can conclude that schizophrenia and epilepsy is disease, or it is some trait which depend on the hereditary and on the environment. Or in another word, we say that this disease have hereditary predisposition. Or another case, measles, concordance of monozygotic twins is about 59, and of the dizygotic twins 51. And we can see that this trait or this disease depends on environment factors, and you know that measles, the etiological factor is viruses. And the last example, intelligence, concordance of monozygotic twins 91, and concordance of the dizygotic twins 88. And we see that the role of the environment more than hereditary. And as a result, we can say that the person's mind is more dependent on his study and effort than on hereditary predisposition. The next methods of the hereditary diagnostic of hereditary pathology is dermatoglyphics. Dermatoglyphics is a study of epidermal configuration on palms, fingers, soles, and toes. There are different types of dermatoglyphics which can include some different groups. It's called dactyloscopia. It is a study of the epidermal configuration of the fingers. Palmoscopia, it is a study of the epidermal configuration of the palms. And plantoscopia, it is a study of epidermal configuration of soles and toes. There are different fingerprint patterns such as arches, loops, and rolls. The most common type of patterns for Europeans are loops. Arches are more common in Negroid race. Eastern and southern countries have a complex skin pattern. The presence of 5 or more arches is a reason to genetic examination because predominance of the arches is a diagnostic symbol of different diseases such as triploidia, trisomy 80, trisomy 8, tetrasomia 9, polysemia X, XXY syndrome. I hope you remember that XXY syndrome, it is a Klinefelter syndrome, or XYY syndrome, prison basketball team syndrome, and Fragile X chromosome or syndrome of Martin-Bell. How can you see the presence or some changes in the epidermal configuration? It is a non-specific symptom of different hereditary pathology. It can be some cause to find some genetic pathology, and it is a cause to more difficult to use some more difficult methods to diagnose this hereditary pathology. And the last type of the diagnostic of genetic pathology is hereditary pathology modeling, and there are two big groups: is mathematic modeling, using different computer programs to recreation hereditary pathology, and biological modeling, recreation hereditary pathology on animals' models. This method is based on Vavilov's law of genetic homology, and this law you must know. This law states species and genera which are genetically close are characterized by similar series of hereditary variability with the correctness that, knowing the forms within the same species, it is possible to predict the existence of parallel forms in related species and genera. It may seem difficult, but it is very easy. Let's see. People and animals suffer from similar diseases. The disease achondroplasia. Achondroplasia is a genetic disorder whose primary feature is dwarfism. In those with the condition, the arms and legs are short, while the torso is typically of normal length. Other features include an enlarged head and prominent forehead. Some animals also suffer from this disease. For example, such dachshunds with short arms and short legs with normal torso. Dachshunds as example of dogs, and in another example of the kitten, it is a cat's munchkin with the short legs and arms and with a normal torso. So people and animals suffer from similar diseases. Another example, vitiligo. Vitiligo is a long-term skin condition characterized by patches of the skin losing their pigment. The patches of skin affected become white and usually have sharp margins. The hair from the skin may also become white. The inside of the mouth and nose may also be involved. Typically, both sides of the body are affected. Often the patches begin on areas of skin that are exposed to the sun. It is more noticeable in people with a dark skin. As you can see on the slide, cats and dogs also suffer from this disease. Albinism, another example that the animals and the people suffer from the same disease. Albinism is congenital disorder characterized in humans by the complete or partial absence of pigment in the skin, hair, and eyes. Albinism is associated with a number of vision defects such as photophobia, nystagmus, and amblyopia. Lack of the skin pigmentation makes for more susceptibility to sunburn and skin cancers. As you can see, albinism is also common for such representatives of the animal world as a koala, kangaroos, penguins, giraffes, and raccoons. And now we start our second part of the lecture, and this part will be about treatment of the hereditary diseases. There are different groups of the diseases, and first group based on the etiological principle, the second group pathogenic principle, next group symptomatic principle, and the last group is surgical treatment. Etiological treatment. I remind you, the cause of the genetic disease is a gene mutation or another changes of the genetic material. So the treatment etiological will be based on the gene therapy. And there are two ways of gene therapy. The first, replacement of a mutant gene with its normal copy. And the second way, injection of a normal copy of the gene while maintaining the mutant. There are some steps of the etiological treatment. The first, creation of an introduced genetic construct by genetic engineering methods, introduction of a genetic construct into cells with help of liposomes or viruses. And the third step is gene delivery to human somatic cells is ex vivo and in vivo. What it means? Ex vivo gene therapy stages obtaining mutant patient cells, isolated cells, culturing patient cells without the organism, introduction genetic construct with normal DNA into the patient cells, selection and extension of the genetically healthy cells, and returning healthy cells to the patient. This type of therapy is known as ex vivo, without the living organism of the human body. And in vivo therapy, therapeutic gene construct packing into the viruses or liposomes and transplantation to the sick people in vivo, in living organism of the patient. Etiological treatment, unfortunately, is very expensive and not always available. Therefore, treatment is often limited to pathogenic and symptomatic approaches. Pathogenic and symptomatic treatment based on the three main ways. First, diet therapy. Exclusion of food components giving toxic substances. For example, diet with patient of phenylketonuria and galactosemia. People who follow the prescribed dietary treatment from birth may have no symptoms. For example, people with phenylketonuria. Their phenylketonuria will be detectable only by a blood test. The diet requires restricting or eliminating food high in phenylalanine, such as soybeans, egg whites, shrimp, chicken, breast, spirulina, watercress, fish, nuts, crayfish, lobster, tuna, turkey, legumes, and low-fat cottage cheese. Starchy foods such as potatoes and corn are generally acceptable in controlled amount, but the quantity of the phenylalanine consumed from the foods must be monitored. The only treatment for classic galactosemia is eliminating lactose and galactose from the diet. Even with an early diagnosis and restricted diet, however, some individuals with galactosemia experience long-term complications such as speech difficulties, learning disabilities, neurological impairment, tremors, for example, and ovarian failure. To follow a low galactose diet, let's avoid foods with lactose such as milk, butter, cheese, ice cream, and other dairy products. Or another way of the diet therapy is adding the necessary food components to the diet. For example, pyridoxine or vitamin B6 for homocystinuria. Pyridoxine, with a deficiency of pyridoxine, homocysteine accumulates in the body. As a result, hyperhomocystinuria occurs. We can correct this condition by adding vitamin to the diet of this patient. The next way, exclusion of the drugs which hereditary caused intolerance is detected, for example, sulfa drugs. The next way, substitution therapy, compensation for the missing products of the missing gene, for example, enzymes, some blood components for hemophilia. And the last type of treatment is the surgery. Surgery used with a patient with difficult malformation of the skeleton, for example, cleft lip and cleft palate and others. The lecture is over. I will see you on the next weeks, and I remind you that we start our new big cycle is parasitology. It will be very interesting, so see you next week. Have a good day. Thank you.